Well, Thomas, it's great to speak to you today, and we're going to be talking a bit about CNS Voyager. So I guess to get started, do you want to just give a brief introduction to yourself just to set the scene, and we can get into talking about CNS Voyager?
Yeah, sure. So I'm Thomas. I'm the CSO and co-founder of Vibraint, the small biotech company in Denmark, offering both services to pharma and biotech academics, doing whole brain imaging, so for preclinical research. But also now we actually have the ambition of building some SaaS products called CNS Voyager. With pretty high ambitions, so like a lot of things we're going to talk today. But yeah, so for CNS Voyager, maybe the best way to introduce this is to talk about the problem itself, actually,
Yeah.
instead of the solution yet. So I think one of the biggest challenges, in my opinion, right now in neuroscience isn't data anymore. Generating data is not the problem. We're actually, I would argue, drowning in data at this point. It's really making sense of it. And it's not to say that it's not an easy thing to do, but we have amazing resources available, right? We have atlases, whole brain imaging techniques, and increasingly more and more rich, molecular data sets. But the main issue is that they're basically siloed, right? They live in their own little space, and pulling them together into one common space is very challenging, right? So you need really specialized people to do that type of work. So the real question, I think, both for academics and pharma is, how do you basically turn all that information into a smart experiment, basically? How do you turn all of this into something that's useful for you? And hopefully that's where CNS Voyager comes in, because the goal is to bring all these different data sets into a common environment and then building tools around them. So researchers can explore the brain as an integrated biological system rather than having basically piecing all that information together themselves. And in many ways, I think the ambition is to build something similar to Google Maps, but for brain biology. And I think I made that analogy maybe before, but like maybe the best way to put this is, imagine, you just moved into a new city. The first thing is when you open Google Maps, you're not usually, at least right away, looking for like a particular cafe or a street. First, you want to get your bearings. So where am I? Where's the city center? Where are the neighborhoods? You start to see the big picture, and once you have that, you can actually zoom in and get the details, right? So that's really, I think, where CNS Voyager starts today, because we're essentially building that orientation layer for brain biology, where research will be given that whole brain view, right? Bringing different types of information into the same space and helping them see patterns and identify where they might want to focus next, basically. And then the vision is to make that zoom increasingly powerful, basically, as the time goes. So once you've found the neighborhood you're interested in, you should be able to go down to, hopefully to follow that same, analogy, the street, or eventually even the specific shop you want to go to, right? So I think in biological terms, that means basically moving from whole brain to a particular region, to specific cell populations and genes, and ultimately to very practical questions about how you might design your next experiments, basically. So the goal isn't just to build another beautiful visualization tool, and that's quite important, right? Because it's really to help researchers make better and faster experimental decisions. That's my dream, at least, right? So we don't want scientists spending most of their time finding and piecing together information, but instead we want them to be testing ideas.
I think it's a really exciting project, and I can't wait to see what this is actually going to look like. But I would actually like build upon like the Google Maps analogy, and I would say this is more like Google Earth, because in
True.
Google Earth you have all of these layers of information that you can add atop the kind of Google Maps view of just the streets and locations and whatnot. You can like aggregate different layers, whether it be socioeconomic overlays, things like that. I see CNS Voyager as a more like a Google Earth, I think, for the brain. But you've explained the concept very clearly there. But what does this visually look like as an interface? It's a platform, a visual platform, right?
Yeah, exactly. So the core of the product, right, is that the centerpiece is a virtual brain. So it's an average of many hundreds of different brains that were captured using the technology we have, and all of this has been aligned to some of the reference atlases that are available. So essentially, you have the brain as the centerpiece of the product that you can interact with in 3 dimensions. And again, you can look at currently, as I said, it's a shallow layer, right? So we would be looking at to go back to what you said with Google Earth, right? We would be looking at the initial layer, sort of like a teaser of what's coming. And that would be, you can look at anatomical brain regions, zoom in, zoom out, see locally where certain genes are expressed, where certain cells live in the brain. And the idea, exactly, the ambition is that this is sort of step number one across many, where we would then be building specialized tools to zoom in and be able to interrogate very precisely, right? What is actually going on here? What are the constituents of that specific sub-area of the brain? How is this brain area interconnected across the entire organ, right? And but more importantly, to interface this information together with your own data, right? So when you bring in, and it could be any type of data, as long as it's data that you can actually bring in and align to that reference space, you can interrogate it inside the tool. And so just to go back to what you said, the core of the tool is very visual, because it's a 3D viewer. But down the line, the idea is to integrate tools to also do statistics and show actually quantitative metrics for you to, sort of explore the diversity of what the mouse brain for now is. Yeah.
And where does this sort of sit on the continuum between, like, being you said it's not just another visual interface, but what you're doing here, for example, taking these brain atlases, overlaying things like gene expression and potentially some other layers, like, this is extremely complex stuff. And obviously the registration that's happening between these different data sets is really complicated. So how far can you trust it for hypothesis generation as opposed to visualization along that kind of spectrum?
Yeah, so I think maybe then it's worth to discuss about the data that is in there, right? Because
Yeah.
how do we actually even generate these maps? What are you looking at, right?
Yeah.
So I think one of the things, as you said, is one of the main features that we will offer from day one is to look at gene expression. And I think the basic idea is to make gene expression explorable continuously in 3 dimensions across the entire brain, right? And this is no magic. So the important thing to understand is that the original biological data that we are using to make that visualization is not continuous. So they come from measurements of individual cells, and each with particular, molecular identity and spatial location. It comes from basically sectioning, making very thin sections of the brain and mapping them back into the 3rd dimension. So you're starting to see millions of discrete cellular observations across the entire brain, but they are inherently discontinuous, right? Because they're coming from 2-dimensional sections. So what we've done computationally here in-house is to use those observations to reconstruct a continuous representation of the underlying biology that we can display throughout the 3D brain. And I think that's a very important distinction when interpreting these maps, because you're not looking at microscope images where every colored voxel corresponds, to something that was directly measured by a microscope at that exact point. But instead, you're looking at the underlying data cellular data that is being experimentally measured, right? But the continuous landscape you see in the viewer is a computational reconstruction based on those biological measurements. So I think so. From there, I think the easiest way to understand it is to imagine that you have millions of dots, right, scattered across throughout the brain. Each of those dots represent a cell, and we know that we know where these cells are observed, and we have the information about their molecular identity. That's essentially our starting point, right? That's the starting data. But what I would argue, looking at millions of individual dots isn't necessarily super useful, right? And importantly, if you looked at another animal, you wouldn't expect to find exactly the same cell in exactly the same physical coordinates in space. I mean, there's natural biological variability from one brain to another, right? But what tends to be much more consistent is the broader spatial organization, right? So a particular cell population might reliably be more abundant in one part of the brain and sparse in another, even though the precise location of individual cells vary, of course, between animals. So what we really want to capture here with that tool isn't every individual dot, so to say, but it's the underlying spatial pattern they collectively form, right? So maybe I can give you a funny example because I was thinking about this the other day. So I'm a big mushroom picking enthusiast.
Oh, really?
And it's yes, and it's right now it's chanterelle season. So from experience, like I know that finding chanterelle is very associated with birch trees, right? Like if you find birch trees, you have good chances of finding these mushrooms. So if I want to maximize my chances to finding those mushrooms in a very big forest, right? What would be very useful, obviously, is knowing where the birch trees are concentrated, right? I don't particularly care about where the exact coordinates of that one birch tree. What I care about is the pattern, right? Is where the birch trees are abundant, where are they clustered, because ultimately that's the information that will help me decide where I need to go and find the mushrooms, right? So I think that computationally speaking, we're doing something similar, but with cells, right? Where we don't care necessarily about the individual cells per se, but we're using really the millions of individual cellular observations to learn the broader spatial distribution of different cell populations across the brain, and a pattern that is more likely to generalize across animals than be like precise positions of individual cells. And we can represent that information, of course, across a three-dimensional grid. This is what people will be seeing in CNS Voyager from day one, but it will be as a smooth voxel-wise map that people can explore in the product, right? So we're essentially going from millions of individual dots to variable observations to a representation of a broader, more reproducible biological landscape, basically. And one that helps you see what you might want to start looking at. Yeah.
That was a really good analogy. I like that, actually. And building upon it, I guess, what are some of the scientific questions that you could see people trying to answer with the CNS Voyager platform?
Yeah. I think there's many, and I think that I think it will depend a lot based on who you are, right? So if you're an academic, for instance, and I don't know, I can give you an example. You're studying like a specific behavior, or let's say feeding behavior. You have data that shows you that there's many areas of the brain that respond to a feeding challenge, right? The mouse is hungry, and many brain regions light up to the mouse being hungry. This might be exciting at first, right? It's a great result. You maybe have 15 different sites of action that are potential follow-ups, but you quickly realize that this is actually more a problem because now we have 15 possible ways, things to focus on. The reality is that, again, collecting data is not necessarily an issue anymore. It's really what do I focus on, right? And it's not to say that this will be the ultimate solution. It's more that the platform should help you guide the follow-up experiment, right? Where it could tell you, for instance, okay, these 15 different brain regions are known to be interconnected. Maybe you're actually looking at one coherent circuit responding to a challenge, right? Then what is the source? Where is the, what is the first brain region responding to the stimulus? What is the output, right? What is the actual end node of the circuit that makes sense for the biological effect that can dramatically help you understand, what is maybe more worth focusing on, right? What is more important for eliciting the effect, right? If you're pharma, perhaps you're less interested into more these more precise questions, but instead you have a clear target in mind. Maybe you already know what you're interested in, like a specific receptor that you're interested in targeting. Then maybe the question is more about where is it distributed? Is the receptor very deep in the brain, located in areas where maybe bringing in a molecule and acting on that receptor will be challenging because you need to cross the blood-brain barrier? So that could be one idea, right? But of course, also in terms of like actually manufacturing drugs, right? If you have a drug and you want to understand where it acts in the brain, you can not only visualize nowadays with some of these techniques where the drug acts, but more importantly, to go a layer deeper in the understanding of how the drug works, meaning what cells respond to the drug. Are you mostly looking at maybe a side effect or, like the direct effect of the drug? I mean, these are very important questions at different scales that people will hopefully be able to answer or at least facilitate, right? Within the viewer. So again, the idea is not really that you will answer precise scientific questions for you, but more to help you guide the follow-up, the best follow-up experiment and most impactful experiment that you can do from the given data that you have in your hands currently, right?
Do you have expectations about how you expect academic researchers versus pharma and biotech companies to be using this kind of platform?
Yeah, so I guess I can give you a concrete example, right? So imagine you run a whole brain experiment, right? And again, you discover that treatment has a significant effect in 15 different brain regions, right? So great. As I said, this sounds fantastic at first, but then reality hits, right? You have too many leads to follow up on. Where do you start, right? So are all 15 effects equivalently important? Are some of them simply downstream consequences of activity that you see somewhere else? Are several of these regions interconnected in actually different pieces of the same biological pathway? Or is there a common cell population or receptor that links them all together? And ultimately, which maybe observation, is going to give you the most meaningful experiments to pursue? So that's where the challenge really begins, because the whole brain experiment has given you an incredibly rich pattern. And this is, to be honest, now something that's happening more and more, right? Data is not really the problem anymore. It's the interpretation, but it hasn't necessarily told you how to interpret, that pattern or what to do next. So traditionally, answering those questions might mean that you move between, papers, atlases, connectivity resources, molecular data sets, and of course your own experimental data. But at the end of the day, all of these are manual, right? So you go and you manually piece together a biological story, and that's science. But there's also real art to it, right? Deciding which connections are meaningful, which observations matter most, and ultimately which hypothesis is worth pursuing. As I said, this is still, I think, more an art than a true science to some extent, right? And what we'd like the tool to be able to do is help those observations condense those observations into a common biological context, so that you can make these decisions much more quickly and have a overview immediately. Maybe you discover that several of those 15 brain regions are, as I mentioned before, interconnected. Maybe they contain related cell populations or share common expression patterns of a particular receptor, but maybe they initially looked like there were 15 independent effects and actually represent different parts of the same circuit, right? And that gives you a much stronger idea of whether the effect might originate where it ultimately leads and which parts of that pathway would be most informative to investigate experimentally as a follow-up, right? So the goal isn't simply to tell you more about the 15 regions per se, but is to help you prioritize them and understand how they might fit together, right? I think that's really the transition that we're interested in accelerating. It's like from seeing a complex brain pattern that, to be honest, no one is able to interpret on the fly, to deciding what it means, what matters, and what is what experiment you should run next, basically. I think that's the most important thing that the tool can bring to the table here.
And to switch topics a little bit, talking about the availability of this on and the features at launch. Can you talk a bit about what features and visualization components will be available when this launches, and then maybe some roadmap pipeline thoughts on what's to come?
Yeah. So I think, as I told you, right now, of course, this project is extremely ambitious, but I think that right now we're mostly asking the researcher to do the navigation, right? So you look at one data sets, then another, recognize relationships, and gradually build a hypothesis yourself. My hope is that what we create is the possibility to change those interactions long term. So imagine being able to come to the platform and simply say, these are regions affected by my drug. What do they have in common, right? Or these are cell populations I'm interested in. What distinguishes them, and what are the most promising ways I could target them experimentally, for instance, right? In very concrete terms, instead of the researcher manually interrogating 10 different layers of data, it could potentially help search across those layers, identify relationships, and prioritize possible explanations. And I think prioritize is really the key word here, because the goal isn't for any of this to replace scientific reasoning by any mean, right? Or tell you what the answer is, but it's really to help you navigate like the biological search space that's becoming too large, to be honest, for any individual researcher to explore manually. It's just not feasible anymore, right? So ultimately, I think that the really exciting combination is the atlas, the researcher's own data, and potentially also AI sitting on top of both, right? Helping turn that information into testable hypothesis. And concretely, the features that will be available now will be, as I told you, the 3D navigation, and also you can switch to a 2D navigation mode where you can also look at a little bit more precisely what is going on. But this is really like a first flavor of what's coming, because I think the most important for us would be to collect feedback on how does that navigation feel, right? The initial the version of that tool, because if the navigation is good and people are happy with how the UI works, then building on top of that, using that as the sort of the solid base to build on top of it is fantastic, right? Then building all these more intricate tools as to maybe selecting subregions of the brain, or like integrating your own data, interfacing with other data sets, extracting statistics, running maybe hopefully even analysis inside the viewer itself, right? Through the cloud. All of these are things that are I would love them to come, of course, as soon as possible, but it takes time to build these things.
Yeah. And so to clarify, is it going to be possible for people to upload and overlay their own data to start with, or is that something that will be further down the line?
Yeah, that will be further down the line. And there's two things really important to separate here. So because we also, part of the business, right, is also services. So if you run services with us, right, so meaning if you actually send us tissue, we run the experiment for you, we do all the whole brain 3D imaging for you, then that data will natively be available in the viewer for you to visualize, but also to overlay all the different data sets that we have available in CNS Voyager. So it's almost like these two will live together, right? It's just that you will have your own personal space inside CNS Voyager, where, of course, you have your own proprietary data, with the addition of all the data available in Voyager itself. And then there's the subscription part, right? So where you're not interested in the services at all, you just want to look at the data itself. There, for now, it will only be the navigation layer, right, where you have access to that amazing breadth of data. But with sort of minimal tools right now to interact with, it's mostly like navigation, right? But again, now the top priority moving forward after that initial launch will be to build some of these features and prioritize features moving forward. Yeah.
Excellent. And if there's one thing that you feel is really important, the take-home message for what people should remember about CNS Voyager, what do you think that is?
Yeah. I think I'd come back to where we started, actually. So neuroscience doesn't really have a data shortage anymore. I think this is super clear, right? And it's getting more and more a problem, actually, also in terms of storage. We have an interpretation problem instead, right? So we can measure the brain at extraordinary scale and resolution. The challenge now is connecting those measurements in ways that help us decide what matters and what experiments to run next. And this is far from being trivial, right? So that's what we're trying to do with CNS Voyager, is make this enormous amounts of brain data something scientists can actually navigate and use, instead of just sitting on a hard drive, right? Because ultimately, I think we don't want researchers spending most of their time looking for information, but instead I would love them to just be testing new ideas, right? That is, I think, the main message I would have to give, and I hope that CNS Voyager can fit, that vision.
Brilliant. Well, thank you very much for sharing some insight and some exciting news there. Been a pleasure.
Yeah, nice.